this screen. So hopefully you can see my okay. So can you see my slide? So now we have about 24 students.
Yes, Doktor.
Najmi ada kat sini? Najmi ada tak dalam ni?
Macam tak ada, Doktor.
Hai, macam mana ni? Farhana boleh tak screenshot all the participant ni? Boleh tak? You nampak tak people yang ada dalam ni? Can you see the name? Farhana? Boleh tak screenshot all the 25 ni? And then letak dalam group tu?
Saya, Doktor Fatin.
Fatin pun boleh.
Okay. Boleh.
Nabila boleh buat?
Boleh.
Alright. Masukkan dalam group supaya I boleh tahu siapa yang hadir dan tak hadir hari ni.
Okey, Doktor. Sekejap.
Tengok Nazmi masuk lambat. Kenapa tu Nazmi masuk lambat? Alright. Just screenshot the names and put it in our group, yeah? Alright. So today is we start with a new topic, yeah? So hopefully you can see my my slide. Okay. This is topic 5, and topic 5 include the rest about the and this is the lecture outline lah. Lecture outline. We start with the phylogenomics. Okay, phylogenomics, metagenomics, and population study. So hopefully all of you can see my slide. Nampak?
Boleh, Doktor.
Alright. So can before we start, can we have doa? Can we have doa?
Boleh, Doktor.
Alright.
Bismillahirrahmanirrahim. Allahumma aftah alaina himmatika wa anshur alaina min khaza'ini rahmatika ya arhamar rahim. Amin ya rabbal alamin.
Okay, so this will be the last applications of functional genomic class lah. Okay, so I think some of this topic will be redundant with your bioinformatics class, but it is important for you to know this is a part of functional genomics. Okay, why we do sequencing is basically because we want to do the comparative genomics, like what we have discussed last class. And for functional genomics, molecular evolution is more on the comparative genomics basically, yeah. Why? Because in this lecture I will explain to you what is the connections between the variations, gene variations, and then how to classify them, how to how to group these among all these species, among all the samples in one group based on their characteristic and the variations, similarity and dissimilarity between them. Alright, so we will talk about phylogenomics, metagenomics, and the population study. Alright, so for phylogenetics, okay, so for phylogenetics, yeah, basically as I said just now, it is a system or techniques you to show the evolutionary history. Evolutionary history means we want to see any variations among all the samples. Yeah, how long it takes one particular individual or one particular specimens to evolution, to have that evolution. Okay, so population genetic is basically is a study of the genetic composition of population, including distributions and changes in genotypes and phenotypes. Okay, in genotypes and phenotypes. Genotypes is what? Changes in genotype means the variations between the genes, yeah, between genes that, or something happens to that genes. So we want to look at that, and as well, uh, we also want to look the relationship, the changes between the genotypes and phenotype. Phenotype means something that you can observe using your naked eyes. Yeah. So we are not just want to discover the changes, but we also want to look at the response to the process of natural selections. Remember yang I cakap last class. Okay, in last class I ada sedikit I've told you about how this we want to see the evolutions, the changes affect the fitness of some individual or some organism. Okay, so that's why we are not just looking at the variation or the mutations that occur to that population, but we also look at the natural selections, the process of natural selections, genetic drift, mutations, and also gene flow. And phylogenomics. What is the difference between phylogenetics and phylogenomics? Phylogenetics depends on regions, small regions or variations in genes. Yeah, but for phylogenomics, is basically they use the characterizations in genomics to and do the classifications. Okay, the purpose of phylogenetics or phylogenomic is to classify what group. Okay, based on the same characteristic, they put it as one group. Okay, so how to classify this? So phylogenomics is based on the genomics. Phylogenetics is based on region, specific region. Contohnya macam as I think you are quite familiar now with 16S. 16S we use that regions for bacteria. Kan? CO1, COX1 ataupun cytochrome oxidase 1, we use that for eukaryotes. Okay, ITS for what? METK for plant. Fungi, ITS kan. So human, for some particular human or for some particular organism, we use the whole genome. Okay, so we call this area as phylogenomic. So the goal is sama. Okay, the objective is still similar. Okay, because we want to see the group that have shared the same characteristic. Okay, shared similarity among the population or among the samples. Alright, so the fields of bioinformatics that integrates knowledge, about the evolutionary history of organism with structural and functional analysis of their genomes, genetics, genomics or proteomics is under this phylogenomics lah. So the goal is to provide an objective measure of the reliability of assumption made about similar genes or proteins in different species. So